Thank you for joining the seminar today. I'm excited to see you all. And I'm also very excited to introduce Yusuf Roohani with the talk, Building a Virtual Cell, an AI Platform for Engineering Cellscape. Yusuf Roohani is a machine learning group leader at the ARC Institute and a visiting scholar in the Department of Computer Science at Stanford University. His research explores how artificial intelligence can guide experimental design in biological discovery.
Sorry, we had a Wi-Fi issue. Okay. All right. As Andre mentioned, I lead a machine learning group at the ARC Institute. For those of you that may not be aware, the ARC Institute is a nonprofit biomedical research organization. We're based out in Palo Alto, right off of the Stanford campus. And my group is working on this effort, which we call the Virtual Cell Initiative. And our goal is really to use artificial intelligence to create better models of cell state and to use these models to predict how cell state may change under different interventions, in particular experimental conditions and perturbations that we haven't actually tested in the lab, but we can predict the outcomes of in silico. All right, so just a brief background on myself. So I've been in this space for about a decade now. I started working on ML for biomedical research and drug discovery broadly in 2016, working at a startup in Cambridge, Massachusetts, looking at modeling network biology in cancer cells, trying to understand resistance mechanisms to chemotherapy. I then moved on to GSK, where we continued trying to build better models of cellular phenotypes, in this case, working with high content screening data, so looking at image-based screens across millions of drugs. I then moved over to Stanford, where I was advised by Yuri and Steve as part of the Stanford AI Lab. That's my son. Here we were looking at kind of moving more into the transcriptomic space and thinking about how we can create better models of cellular transcriptomic state and trying to understand how that would change under interventions, in particular looking at combinatorial interventions and how we can model that using a variety of approaches ranging from graph neural networks to large language models. And then since last year, I've been leading a machine learning group at the ARC. And here we are essentially trying to take those ideas to the next level by scaling up both the data generation as well as the model development, and trying to see how far can we push these ideas. What does it take to really make models that are actually useful within the lab? And so it's been great to kind of think about not just the model development side, but also kind of data generation and how we can guide that. All right, so moving over to the science, I'm sure I don't need to convince anyone here that AI has transformed scientific discovery. My favorite example is AlphaFold. So AlphaFold takes in a protein sequence and will predict for you the three-dimensional structure of that protein. And so essentially it's learning this mapping from sequence to structure. And by being able to learn this representation, it can then predict how other proteins would fold, even those that it may not have seen at the time of training. So over here, you can see that AlphaFold is able to map out a very large space of possible protein structures. And many of these shown here in blue, it hadn't actually seen in its training set. And if you think about it, experimentally uncovering each of these structures takes a lot of time and effort. It's not an easy process. And so in a way, what AlphaFold is doing here is it is performing this experiment for us in silico. And this kind of template of really being able to perform in silico experiments, map out this large space of possible hypotheses, and then search for the ones that are most interesting. This is a template that we actually find not just in protein structure prediction, but in scientific discovery broadly. And so one way to think about scientific discovery is that it's a search problem over a space of potential hypotheses. And so what we wanted to do was take these ideas from the molecular level and sort of take them up one level to the cellular scale. So can we move beyond modeling individual molecules and individual proteins? And can we now begin to understand better instead how these proteins and how these different molecules are interacting with each other within a cell, which is a much more complex and noisier system. And of course, kind of build up from there to more higher level phenotypes at the tissue level and the organismic level and so on. And so why would this be useful? You can, once you're modeling phenotypes at the level of the cell, it's easier to begin to query questions that might have therapeutic relevance. So for instance, here, if you have a space over neurons, and you're able to identify different forms of disease neurons, such as degenerative neurons, hyperexcitable neurons, and you have more functional or healthy neurons. If you had a working virtual cell model, you should be able to identify what are transitions that would take cells or take these neurons out of these more disease states and move more closer towards healthy states. And that's really our goal here is can we use a model that understands cell state intimately well to perform in silico experimentation. And by doing so, also accelerate the discovery process because you have more likely hypotheses that you can then drill down on and perform in the lab. Another aspect that becomes apparent through this description is that it's very important to be able to connect these models back to experiments in the lab. That's something we've taken very seriously, and we wrote a perspective about how we think you should build the AI virtual cell, which was published in Cell last year. And we kind of described this idea that these models cannot kind of sit in isolation and cannot just be developed without any consideration in terms of the data that's being used to build them, and also without kind of without being employed to generate the next set of data to further empower these models. So really, it's kind of this closed loop where you have these virtual cell models that are able to help you understand cell biology in silico, propose hypotheses, which you can then test out experimentally in the lab, and then feed back into your model to further strengthen it and further advance its understanding. So this is our vision. And so in the rest of this talk, what you will see is that we never really think about the virtual cell or this vision of the virtual cell as just a model, a predictive model in isolation. We also think about the data that goes into it and how we can more strategically generate the right data to power and strengthen these approaches downstream. And so that kind of segues nicely into the structure of the talk today. I'll be talking to you about the work in my group, which kind of focuses on these four broad themes. On the left here, what you see is, as I mentioned, how we're leveraging AI to build better models and also to create better evaluations that connect back to what is more relevant to the biologists, to the clinicians, to other people using our model, and also how we can leverage AI to generate data to power these models. And so I'll describe some work where we used AI agents to curate vast biological data repositories that can then be used to train these models. And I'll also describe, if we have time, some work we did in using agents directly to design genetic perturbation experiments in order to further optimize and arrive at phenotypes of interest. So over the past year, we've had papers that we've put out in each of these areas. And so just briefly touching on each of them, I'll talk to you first about state, which is a transformer-based model that leverages data from sets of cells to better predict how those cells will respond to perturbation. And this uses single-cell transcriptomics data. I'll also describe the virtual cell challenge, which we launched at the Arc Institute 3 months ago. It now has over 1,000 teams competing to create better models of cell state that can predict how cells will behave under intervention. I'll also talk to you about scBaseCount, which is an agent that we developed to curate single-cell transcriptomic data sets. And at the moment, we've created the largest existing single-cell data repository. It's over 3 times as large as the next largest repository of single-cell data. And lastly, I'll talk to you about Biodiscovery Agent, which is another agent we developed for being able to guide and design perturbation experiments. All right, so I'll pause there. If there are any questions right now, I can answer them. Also, feel free to stop me in the middle if you have questions or if anything is unclear.
Can you
Hi, can you slow down a little bit?
Okay.
for the accent, yeah, that more probably go. Sorry.
Oh, sorry, I didn't realize.
Thank you.
I thought it was only on my screen. Okay, great. Let's keep going. All right. Okay, so let's first talk about state and really what the goal really here is, right? So before talking about like virtual cell models or even just cell state models, whatever you want to call them, what really is the goal? Why do we even call this the virtual cell? Well, where does this come from? So this is not a concept that we created. It's not a concept that even is very new. It's actually something that's existed in the literature for a very long time. For those of you that may have heard of this field called systems biology, it essentially had this very same idea that can we create mathematical models of cell behavior and essentially train them on experimental data that we generate in the lab and then use that to predict outcomes for new experiments that we've not done before. In the past, the way that they approached this was by developing systems of differential equations. So there's seminal work by Oriolon's group where what they did was they mapped out the reaction kinetics between different enzymes and different proteins within the cell. They created a system of differential equations. And then the goal then was to be able to tune different parameters or change the input settings for this model to be able to predict what would happen were we to perform a different experiment. There was also great work from Marcus Covert's lab at Stanford, which created what they call the first whole cell model. And they tried to map every single interaction within the cell across DNA, RNA proteins, and various different cellular components, resulting in over, I think, several thousand differential equations, which their solver would solve for simultaneously to try to predict what would happen to the cell under, a different input or a different condition. But so these models are very useful. They definitely informed a lot of our understanding of the biology of these specific cells. But as you can imagine, they don't translate and they don't generalize very well beyond the specific cell or the specific context that they were trained on. And so we wanted to explore how can we leverage more modern approaches to machine learning and artificial intelligence to further enhance this process and leverage the large amounts of data that we're generating right now to kind of more learn these relationships in a more unbiased perspective. And so here comes in kind of the foundation model approach. So as many of you are aware, artificial intelligence has, there's been a lot of developments in the AI research area. And I think one big cause of this has been this foundation model approach of looking at machine learning. And what this means is that you no longer design your model specifically from scratch for a specific task, but instead you build what are called these foundational models that are trained unsupervised on large amounts of unlabeled data. So this could be text, it could be images, it could be speech. And then what you do is you then, after this model is trained, you apply it downstream to various different downstream tasks, usually through some form of fine tuning or other adaptation. And many of these tasks, such as, for instance, answering questions or captioning images, may not directly be trained for in your pre-training process, but you can leverage the same general representation downstream for a variety of different uses. And so we wanted to kind of bring this same approach to the modeling of data from the cell. So can we learn these general purpose representations of cell behavior and then kind of apply them to various different downstream tasks to assess how well the model is working? All right, so of course, there are many different tasks that would be of interest. In particular, what we were really interested in was being able to predict the effect of perturbations. So the specific formalization that we looked at was given a distribution of unperturbed cells, say you apply some perturbation to these cells, what is the distribution of perturbed cells going to look like? And essentially, we wanted to develop models that are able to learn this effect. So can we learn the distribution of perturbed cells given unperturbed cells and an applied perturbation? Of course, there are various other things that might be of interest when you're studying cell behavior, but for us, this was something that seemed very central and very important for really understanding the regulatory structure of a cell and how it functions and how different genes interact with each other. Yes.
Sorry, just one question. Are you are XP and X naught population level?
Yes.
Or individual cell level?
Yeah, that's a good question. Over here, it's kind of loosely defined, but you can think of it as a population.
Can you repeat the question, please?
Yeah. All right, so how are we thinking about X naught and P? So of course, there are many ways of thinking about cell state. In our research, we focus a lot on transcriptomics, largely because we have a lot of data at the single cell level for transcriptomics. But of course, there's also a lot of exciting data in imaging, proteomics, cell viability, which can also be explored in future. And so we in particular looked at a few different data sets that were very interesting. So as I mentioned, BaseCount, which has over 500 million cells worth of data from 70 different tissues. We also made use of data from the Tahoe 100 data set, which had about 50 different cell lines. And of course, the cell by gene corpus, which also has data from various different tissues and species. All right, so in terms of perturbing cell state, we looked primarily at genetic perturbations. In particular, we were very interested in using perturb-seq data. Again, large cell numbers, large number of perturbations, kind of easily scalable. And so this was like a very relevant data type for us to teach the model some causal structure around what happens under different interventions and how the transcriptomic profile is impacted. All right, so what is the machine learning task more specifically? So say you have a given cellular context, you have a certain number of perturbations that you've already tested, and you know what the cell state is in response to those interventions. You're then interested in being able to predict what's going to happen for a new unseen test context. And we call this the perturbation generalization condition. This has been studied quite extensively in the past. So what we were more interested in is actually a slightly different variant of this task. We were interested in essentially not just looking across perturbations, but we were more interested in looking across contexts. And for us, this was something a bit more tractable and also something that can actually be quite biologically useful. So for instance, if you have data from a couple of different donors, say you have T cell populations from 50 different donors, and you know how those T cell populations react to various different perturbations, you know how the state of those T cells or their activity varies, and you get data from a new patient, we want to be able to build a model that could then predict for this new patient, given the data that we have, given the transcriptomic information that we have or other variables, can we predict what would happen under the same set of perturbations? And so that's the setting that we thought would be more useful and is in fact, from a machine learning perspective, more tractable. And so that's what we focus on a lot more in our model. All right, so again, this is not something that is new. This has also been studied in the past. And so a lot of existing models, what they do is they will take one cell at a time from each of these two distributions. So you have your input cell distribution, your output distribution, and they try to learn a model that can predict perturbation effect conditioned on the specific donor that you're trying to make or the specific cell type, however you're defining context. But what we found is that a lot of these models are not able to perform very well against even and will often not outperform simple baselines. There have been a lot of papers within the past 2 years, within the past year actually, that have evaluated existing models and found that they often will fail when you compare them to linear baselines or just predicting like the average effect. And we believe this is largely driven by the fact that existing models are not meaningfully accounting for the strong heterogeneity that exists within these populations. We think that it's important to model the heterogeneity both within a specific cellular population, but also across different experiments. And so in our model, we're really focused on specifically modeling those effects and allowing the model to learn the true perturbation effect, really kind of disentangle that true perturbation effect. And we found that that helps us quite a bit in performing better than existing approaches. And so what I mean by that is if you have this unperturbed population and a perturbed population, you can model this as essentially this perturbed state can be modeled as composed of three different factors. You would have the true effect, that is the true effect caused by the perturbation. Then you have the heterogeneity within that basal population. So say you have cells in different phases of the cell cycle. Each of those might respond slightly differently to that same perturbation. And so we want to be able to model this unannotated variation within the source population, and also the noise between different experiments. So if you're combining data from various different perturbation experiments or single cell experiments, you might have different sequencing depths. You might have performed the experiment under different conditions. So how do we model that effect independently as well? And so we create a model that essentially accounts for all these three factors. So this is accounting for the heterogeneity in the source distribution. This is accounting for the true perturbation effect, and this is accounting for the technical noise within experiments. Yes.
Sorry, is this a qualitative equation or a technical equation?
It's still fairly qualitative, but we do map it back to specific parts of the model in the next few slides.
Okay.
So, yeah.
I'll wait.
Okay. Yeah, but happy to answer questions after that.
Okay.
Yeah. So what we did was to resolve the within dataset heterogeneity, or the first two terms in this equation. We came up with this idea to basically train a transformer over sets of cells as opposed to individual cells. And what this lets our model do is essentially by visualizing not just individual cells at a time, but instead looking at like large samples of cells from this distribution, it kind of intrinsically begins to learn what is the variation between cells within this population. How should I model that? And so the way that we do that is we first stratify the input population by known covariates. So if we know what cell types are in this population, we know what perturbations are applied, what experimental batches are present, we'll stratify that population by each of those different covariates. And then what we'll do is we'll pair up the unperturbed and perturbed populations with matching covariates and use a maximum mean discrepancy loss, which is a distributional loss, to align the predicted perturbation effect and the true perturbation effect. And so what this does is it doesn't enforce that the model learn a one-to-one map between cells, It allows the model to essentially leverage the full distribution of cells to learn an effect. And at the same time, it also begins to capture some of that underlying heterogeneity within the basal cell population. So in more implementationally, we developed this transformer, as I mentioned, where every token within the input sequence is actually an independent cell, as opposed to being independent genes, which is what you'll see in a lot of existing models. And you predict how each of those tokens vary following the transformations within the model. And this gives you a sense of what the predicted output expression would be for that distribution of cells. All right, and then for the epsilon term, we wanted to be able to model different data sets. So when we're training our model on multiple different data sets, we make use of an embedding model. And what this embedding does is essentially, I won't go into details here, but we optimize the model to focus on true perturbation effects and reduce the effect of noise between different experiments. And this allows us to more effectively train across different settings. So what does this look like? So here you can see how the model's performance varies as you increase the cell set size. So on the y-axis over here, what you're seeing is the validation loss, which tells you how effectively is the model able to predict that perturbation effect that you're interested in. On the x-axis, what you're seeing is the amount of compute that you've used. So it tells you how many operations the model has, how many operations have been performed during the training process. And as you can see here, as you increase the cell set size, as the green gets darker, the loss for the model, the validation for the loss of the model keeps decreasing up until a certain point around 256, at which it starts to plateau. And beyond that, the model doesn't get a lot better for this specific data set. But the core idea here is that even though you might see the same number of cells and you might perform the same amount of computation, if you're able to see multiple cells at the same time during a forward pass, the model has a better understanding of the underlying distribution, and it more quickly learns to predict perturbation effect more accurately. All right, here's your question.
So you answered my other question.
Yes.
We carried slides already, but I have a new question.
Okay.
Sorry. So am I right in thinking that what you're doing here is the model is, you're not changing the input and output of the model as compared to some of the prior approaches. You're using this stratification and maximum discrepancy to restructure the loss. Right. That way the model is sort of getting a better learning signal without making the model take in a whole population of a certain size as input. Is that correct?
Yes, yeah. So what we're doing, exactly. So a lot of existing transformer-based approaches in the space would take individual cells at a time and try to model effects. But what we're doing is giving the model visibility to a large set of cells. So almost like a large sample of the population. Yeah.
But the model can still operate on, like at inference time, the model could still take a single cell as input.
Yes, it could.
Okay, yeah.
It could, yes. Yeah.
So in one of the previous slides, you said that you treat entire cells as tokens, right?
Yes.
Is this, do you feed in like a vector of the entire RNA expression profile, or do you select certain RNA expression features? And if so, how do you do that?
Yeah, no, that's a great question. We have two versions of our model, one that works directly on gene expression and one that works in the embedding space that I mentioned earlier. So when we have like this version that works on the embedding space, we actually embed the RNA profile of the cell into a lower dimensional embedding of about it's, I think 256 or 512 dimensional embedding. And that is fed in as the token. But in the case of gene expression, what we do is we, at the moment, we'll take the 2000 most highly varying genes, and we will pass that through an MLP that creates a lower dimensional representation that is then fed into the model as a token. So it doesn't have like gene level like dimensions, but you can, if you really want to, you can map back to. Any other questions? Okay, I'll keep moving. And we could also ask, like, are you actually, I mean, what if you did something simpler without using self-attention? Say you just fed in through the transformer the average. So you just tried to use the pseudobulk expression as input. That's what you see as the solid gray line. It does quite well, but it's still not able to outperform the model that's seeing a large set size as input. And you could also provide as input the same cells, but not provide any self-attention. So not make use of the transformer's attention mechanism. And that doesn't do as well. So really, both the cell set component as well as the self-attention help the model learn new aspects of the underlying cell's heterogeneity and the perturbation effect that is otherwise lost when you remove those components from the model. Yes.
That's off to the box. versus
Oh, you mean like randomly sampled pseudobugs?
For example, each of your given single cell will be
Yeah.
neighbor of 100 cells.
Yeah.
Just one sample.
So we've tried that in a different model. We haven't tried it here. I think the challenge with doing that is it tends to it changes the distribution that the model is used to looking at. And then you're kind of only restricted to always predicting pseudobugs. So for some people, maybe that's fine for some use cases. We haven't tried it in this setting. It might actually work. But yeah. All right. Can I take it in a little? Okay. Okay. All right. So if you look at the attention heads, it's quite interesting. You see, I mean, it's not very informative to put too much weight on attention heads because you can pull out any story from an attention head. But what's nice to see is that you do see different patterns, right? So you see some of the attention heads are just like flat vertical lines, which shows that in these cases, the model is learning some kind of a more suitable response across different cells, meaning that it doesn't vary. And then there are other examples where there is a variation across different cells. And so it does learn more cell-specific responses. We also compared our approach theoretically to optimal transport, because essentially what we're learning is something very similar to what optimal transport does, which is it tries to learn a mapping function that minimizes a cost, and you kind of predefine what the cost is, except in our model, we're not explicitly defining the costs. So we have fewer assumptions. We're technically easier to scale, at least in our current implementation. And we found that theoretically, within our space of solutions, we also are able to learn optimal transport, which is quite exciting. All right, so we then spend a lot of time thinking about model evaluation. So another challenge with a lot of the existing perturbation models is they haven't really developed metrics that look across a range of metrics, right? So a lot of them will only focus on Pearson correlation, but it's very interesting also to understand how is the model doing on differential expression? How is the model doing on detecting things that biologists care about? So as a first step, what we did was we developed an evaluation procedure, which we thought resembled a common setting within experimental discovery. So first, we looked at 3 different data sets. We looked at chemical perturbations, signaling perturbations, and genetic perturbations from 3 different data sets. And we set up the evaluation such that the model would have access to a number of different cell contexts or cell lines at the time of training, and it would predict the effect in a held out cell line at the time of test. To make the task a little bit more tractable, we allowed the model to see 30% of the data from that context at the time of training. And this is not something that's not very common in academia or industry. Very often you will run a perturbation screen very deeply in specific cell lines and maybe a more shallow screen on other cell lines. And so this is a kind of recapitulating that same setting. And then the goal of the model is to predict what would happen in this 70% held out test set. In addition to that, we incorporate baselines to make sure that the model isn't just learning to average or copy over effects that it's seen at training time. So we have 2 specific baselines that are strongest in our opinion. The first is the perturbation mean baseline. And so what this does is that for the training context, you essentially just take the average of the effect and you paste it over for the test context. So this would be just simply learning to average what you've seen at training and just pasting that in the test set. Or you can also learn from the 30% that you've seen held out in the training set and just kind of copy that over. And we call this the context mean baseline. We also developed a comprehensive evaluation suite, and this is now being used by over 1,000 teams from across the world as part of the Virtual Cell Challenge. So we're really excited to see how well this has been holding up to a lot of testing and a lot of different users. And the goal for this evaluation suite was how well does state recapitulate a real perturb-seq experiment. And so in our mind, when you run a perturb-seq experiment, one of the core things that you try to look at is expression counts, you look at differential expression, and you look at overall effect sizes. And so we designed metrics to look at each of these, which are to a large extent independent of each other, but also, of course, there's some overlap in what they're measuring. But it gives you a more interpretable understanding of how the model is working. And so we have like a number of different metrics here. I'll walk through some of these in more detail, and maybe I'll just go into that instead of listing them. So first, let's talk about some of these expression-based metrics. So one metric in particular that's been very effective for us in determining how well the model is working is something we call the perturbation discrimination score. And the idea here is how well does the model learn to discriminate between the effects of different perturbations? So for instance, say you have a prediction that your model makes, shown here in red, and say this is the ground truth expression based off of the ground truth data. you want to learn how well does the model do in predicting, how similar is your prediction to this ground truth as compared to other prediction data points in your training set. So the blue points correspond to other true perturbation states. The green is the real state for that specific perturbation, and the red is our prediction for what that state would be. And so what you want is that you want the red and the green to be as close as possible. And so what we do is we use a rank-based metric to measure how similar they are. So we start numbering these perturbations based on how similar they are. So this is the most similar. So this gets a rank of 0, this gets a rank of 1, and this gets a rank of 2, and so forth. And then your score is essentially just the rank that the right ground truth value got as divided by the total number of comparisons that you made. And so the ideal model would always be closest to the ground truth prediction for every perturbation as compared to other perturbations in your test set. Any questions? Okay, so this was a very informative model, and we applied this to a couple different data sets. And you can see that in blue here is the prediction made by state. We compared this to the 3 baselines I mentioned earlier. So you have the perturbation mean model, the context mean, and a linear model. All of them don't perform very well. But they still perform better than the existing deep learning baselines, including SDVI, which is a autoencoder-based variational autoencoder, and CPA, which is also an autoencoder, and SDGPT, which is a transformer-based foundation model. All right. We then moved on to looking at differential expressions. So for those of you that haven't worked in transcriptomics, a differential expression is like the bread and butter of working with these data sets. And the goal here is to look at each individual gene and try to identify where are we seeing significant change in expression following a specific condition, such as treatment with a drug or in our case, a genetic perturbation or other interventions. And so we looked at 3 different aspects of differential expression. So whenever people talk about differential expression, they'll usually show you this volcano plot. On the x-axis here, what you're seeing is so every dot here is a specific gene. On the x-axis is the log-fold change of that gene following perturbation. On the y-axis is the significance of that change in expression. And so first we wanted to see how well are we doing in detecting the significance of change in expression. So on the y-axis here is the predicted significance based of a different model, in this case state or the other baselines. And on the x-axis is the true significance. And you can see that it's pretty good concordance in the case of state as compared to the other models. We also then, you can also look at this across different perturbations. And if you do a precision recall curve, again, we see that state does pretty well compared to the same baselines. And then we also wanted to see like log-fold change. So here we're looking at the x-axis. So if the model predicts that this gene is going to go up by 2-fold change, does state or other predictive models also predict 2-fold change, or do they predict something else? And so here again, we see that state does quite well at capturing the true log-fold change as compared to other baselines. All right, so how do we, well, here's some more examples. I can skip this. All right, so how do we use this in practice? So I've shown you that state is doing a good job of detecting these cell type specific effects, but then how do we actually leverage this for real discovery? So we looked at one specific drug, which is a drug for melanoma. It's called trametinib. And over here, you can see the performance of state compared to 2 different baselines. Again, the context mean, the perturb mean. And then we identified here one of these dots corresponds to the perturbation caused by trametinib. And what we did was we looked at a specific drug, which is a specific cell line C32, which is a BRAF mutant cell line C32. And trametinib is specifically designed for BRAF mutant melanoma. And we wanted to see, is the model actually able to detect gene expression that is specific to this BRAF mutant cell line? And so how do you understand this plot? So here we have 5 different cell lines that were actually kept in our test set. Each axis, each column here corresponds to an independent gene. And so the green corresponds to, here is like the ground truth. So these are all the genes that are differentially expressed in the ground truth. This is for this other cell line. And so you have this for 5 different cell lines. And then here are the predictions made by state. So you can see that state is able to detect a lot of these different different differentially expressed genes across different cell types. And the nice thing is it also detects genes that are agnostic to cell type, right? So for instance, some of these genes over here, they show up in all the different 5 different cell types, and state is able to actually detect those 5, which is detect them across the various different contexts. Yes.
You were asked if you could speak a little bit slower so that the
okay.
could transcribe.
Okay, sure, no problem. All right, I'll try to talk a bit slower. Yeah. Yeah, so and this was great because what we're seeing is that for this FDA-approved drug for BRAF mutant melanoma, we're able to see that the model is actually detecting effects in BRAF mutant cell lines that are specific to that cell line. And so this kind of goes back to our initial motivation that if we do build these models and they are really donor specific, we could then hopefully leverage them for designing more personalized treatments and personalized therapeutics. So some more examples of the model's predictive use. So here, essentially, we're looking at state predictive values for cell survival, and we compare that to actual values. And again, we see that the performance of our model is better than if you were to use different kinds of mean baselines. All right, I'll pause there. Any questions before I move on? Okay, cool. All right, so I'll just quickly go through this as well. So really the vision for the virtual cell is to not just to be able to predict effects within the specific data set or the experiment you're performing, but really to be able to leverage these models across different contexts, across different settings. And so for this, what we were excited about is, can we actually leverage models like state and bring to it like brand new novel cell types, novel cell states, novel donors, and then be able to predict what the effect of that perturbation or of that intervention would be. And so what we did was we actually built we built our own foundation model as part of state to really learn this general purpose representation across these different contexts. And we compared that to existing foundation models like UCE, SCGPT, transcript former, and we found that our model was actually very effective at being able to separate between different perturbations. So on the y-axis here is the model's ability to separate between perturbations in the true data. And on the x-axis, what you're seeing is if you actually use that downstream as part of the state model, you see better performance if you use a state embedding as opposed to using other embeddings. So that was exciting for us. And then we applied this downstream. I'll skip this. We applied this downstream to the setting where we essentially pre-trained our state model on the Tahoe dataset, which had 50 different contexts with over 1,000 perturbations. So we pre-trained our model on that large chemical perturbation dataset. And then we fine-tuned it on various different downstream datasets, including some genetic perturbation datasets. And we found that by using our embedding model and the state architecture overall, we were actually able to make predictions in the zero-shot setting that were quite effective for certain metrics, not for all metrics. In particular, we were very good at predicting the overall effect size of a perturbation. We didn't do as well on predicting differential expression, and that's understandable because it's a much harder task. But it's very encouraging to see that they're using these models that account for heterogeneity across data sets and within cell populations. We're slowly able to piece together the signal from across these different data sets and build these general purpose perturbation models that can be applied to different settings. All right, so the next thing we wanted to focus on is how do we kind of bring this discussion around virtual cells and predicting perturbation effects? How do we bring the community input into this, and how do we kind of really have a larger discussion around what is the best way to evaluate these models? What is the right task? How should we best be generating data? And so in this spirit, we launched the Virtual Cell Challenge a few months ago. And the idea behind the virtual cell challenge was if the community is really excited about the potential for artificial intelligence to impact our ability to model cell behavior and understand cell state, how do we formalize this task in the same way that perhaps CASP did for the protein structure prediction problem? And so we wanted to really start that conversation with the community and ask them, and basically we proposed our formulation of the task. And also now we're getting a lot of feedback from the community on how they think this task can be adapted and improved. And so again, the task that we focused on is the context generalization task in perturbation prediction. We also developed a purpose-built, very deeply sequenced single-cell perturb-seq data set just for this challenge. And it's been very well received by the community. We've been told by many people that it is possibly one of the highest quality perturbation data sets that they've used. And so really excited about how we can kind of take this forward and really become similar to how AlphaFold used CASPs to optimize over the years and become like a really powerful model. We want to use the Virtual Cell Challenge as a launching pad for really strong models that can begin to do very well on this task. The competition has been launched for a while now, and we're actually coming close to the final end date, which is I think about a month away. We've had about 3,000 registered participants. Over 1,000 teams have made submissions already. I think the latest number from this morning was 1,036. So it's been great. It's been great to see the involvement from the community, and it's been great to receive the feedback on what's working and how we can kind of improve this and really steer it towards the most impact for the scientifically and also more broadly. And I also just want to make a small point that the data generation for this challenge was a very long and involved process. We wanted to be very strategic in the perturbations that we selected. We also wanted to really deeply sequence and have very high cell coverage. So the way that we did that was we first ran a low depth screen over 2,500 perturbations in human stem cells. And then we looked at the data, the perturb-seq data for that cell line, and we grouped different perturbations by their effect sizes. We clustered them by phenotypic diversity, and then we carefully picked perturbations that had maximal overlap with existing data sets so that different teams can train on existing data sets. And finally, we ended up with a very large and high-quality data set of 300 perturbations, each with over 1,000 cells per perturbation. Just for context, most existing data sets will only have between 30 to maybe at most 100 cells per perturbation. So this was like almost an order of magnitude more. And we also sequenced it very deeply at 50,000 UMIs per cell. So you get a very precise kind of gene level expression. And so it's been great, and and we're excited to see the outcome and how this impacts the field and how it kind of accelerates development towards better models of predicting cell behavior. All right, I'll pause. Any questions so far? Are we good? Okay, cool. So in the spirit of like clean, large data sets, we also wanted to think more deeply about how we can leverage AI, not just for developing models and evaluating them, but also thinking about how can we better curate curate public data repositories and clean them up using AI. And so in this spirit, we developed what we call scBaseCount. And the motivation here was almost every paper that is published that uses single-cell data, actually every paper that is published using single-cell data, you have to submit the raw sequencing reads to SRA. And so SRA has an enormous amount of data, but it's all kind of like raw raw reads from your sequencer. And so we wanted to see, can we just like directly go into SRA and reprocess all the single-cell data that's in there and essentially create our own kind of very large single-cell data set. And we estimated that there must be over 500 million cells worth of 10x data in SRA, and the largest existing repository of single-cell data is just at about 120 million. The challenge, of course, is that the metadata is a big mess. It's very difficult to process manually, and so that's one of the main reasons nobody's done it. So what we did was we created an agent to do this process for us. So we designed a hierarchical agent workflow to look through every single 10x record within SRA, process the metadata, and then feed that into a recounting pipeline, which essentially would do all the sequence alignment and count all the different gene expression counts and give us a final cell by gene count matrix for every data set. And the goal was to enable better development of AI models downstream. So here's the overall pipeline. So we have SRA agent, which automatically keeps looking for data sets in SRA. It finds them, and then it has other agents that will assign different metadata components to that specific data set. So there's a number of different agents here. I'm just kind of skimming over this, but it's a hierarchical workflow. And then when the data set is ready with associated metadata, it's fed over to our next flow pipeline for performing the recounting, aligning the reads, and then getting the final gene expression count matrix. And so one of the benefits of doing this, of course, is that we uniformly process all the data using the exact same read alignment and the exact same gene sets. So it's a really nice large data set which has the exact same number of genes with expression counts. And so it's very easy to also deal with that data set and also read out not just protein coding genes, but also non-coding genes, also looking at intronic expression, exonic expression. So it's a really valuable data set. So to give a sense of scale, right now, scBaseCount is at over 500 million cells. So it's over 4 times as large as the largest single cell data repository cell by gene. It's also the most diverse. We have over 27 different species, including both plant and animal species. The largest, of course, are human and mouse, where you can see we have about 300 million human cells and about 150 mouse cells. It also is the largest repository that is uniformly processed. So we see actually reduced batch effects. We also see that it's we are also the largest repository of non-coding gene expression. So you can actually see how these non-coding genes vary across various different cellular contexts. And it's also the largest biological data repository, to our knowledge, that has been curated by an AI agent. The fact that it's curated by an agent gives us a lot of power to keep growing this repository. And in fact, it does keep growing. So we just kind of send off the agent to go, look through SRA again and identify new data sets and process them. And so it's a continually expanding repository of data. And here on the left, what you see are the various different cell types and tissues that are represented within this data set. We see an overabundance of T cells and immune cells, but there are also other cells that are represented, such as epithelial cells, endothelial cells. And we've done a lot of analysis. We have an updated preprint that should be out soon as well. So we did some downstream analysis as well. What is the benefit of having a large uniformly processed data repository? We see that the batch effects are significantly lower, especially if you compare, if you try to see how much of the signal within this data set can be explained by technical factors such as sample ID or single cell versus single nuclei or the library prep chemistry, then in each of these cases, in terms of base count, the signal is lower than if you compare it to something like cell by gene. And then we see that in terms of more biologically meaningful variables, such as tissue, a lot of more of that signal can be explained in the case of base count as compared to cell by gene. So it's all kind of very positive indicators that doing this uniform processing helped us get cleaner data downstream. We also use this to train an AI model, in this case state, and we looked at how well are we able to distinguish between different phenotypes, including cell type, disease, perturbation. And we found that in all of those cases, base count was able to do quite well. The differences were less stark in the case of more more easy to separate phenotypes like cell type. But then when we go down to the level of differentially expressed genes, we found that using base count gave us a significant edge in being able to detect variation across different cell types and cell states. All right. Okay, I'll pause here again if there are any questions. Yes.
About how you have a variety of different types
Right.
with your data consortium. Do you have any understanding of how generalizable results are across cell types, or does it kind of depend on the problem?
You mean for our data curation effort or for the model that I showed earlier?
And like,
right.
When you think about scaling these experiments to different cell types.
Yeah. So that's a really good question, right? And I think I had some results at the end where it showed that there is some kind of meaningful improvement, but I would say that still these models are far away from where you can just like point at a cell type or point at a specific tissue and say what's going to happen there, like what is the effect of this perturbation going to be there. I think right now, at least within our model, we find that if you have some amount of experimental data for that condition, for that cell type, it tends to do much better. It tends to like learn the underlying regulatory structure a bit better. Also, so I guess the short answer is that I don't think we're there all the way there yet, but it seems like there's a good sign that we've made good progress. And also, I think at ARC, we think that if we standardize the data generation process, we can probably also make a lot more progress. One of the big issues with these perturbation experiments is that everyone does them slightly differently. And so what a knockdown of a specific gene means to me in my data set might be very different from what it means in yours. And so really trying to predict that is kind of very hard for the model for that's also another reason. But yeah, I think there's a lot of work that needs to be done. Yeah. Yes.
So in the slide where you show the distribution of cell types in your data set, you showed that you had like a very big concentration of T cells and immune cells. And this caught my attention because historically, like immune cells often have, you were mentioning earlier, donor-specific behaviors. And this can be for HLA alleles or very many number of reasons. Does your model learn to, I guess, per donor analyze the effects, or does it learn it across all donors?
Yeah, no, that's a great question. So in our model, as it is right now, we've not dived into this too much, but we have another model we've been working on, which specifically focuses on this. And in fact, we actually find that we do have pretty good generalization capabilities for T cells. I think for this reason, because there's just such an overabundance of T cells in the base count data set. So definitely, I think that does help. It really helps, I think, because I think a lot of studies have been really focused on like cancer, looking at blood, looking at immune cells. So that it doesn't surprise us that like immune cells and T cells are so overrepresented. And definitely for the models that we're training on this data, we do find that generalization is better when you're working with immune cells, when you're working with blood cells. So definitely it is having an impact. Yeah. Okay. I have about 5 minutes, so I won't go into too much detail for the next one, but I'll just do maybe 2 slides. So the last thing we wanted to look at was, can we use these agents to actually tell us what experiment to do? So far we've kind of been using them a bit more passively in terms of here's the data, can you clean it up? Can you help us like, make this large standardized repository? Now we wanted to see, can we actually have this agent design a lab experiment and then take the readout from that lab experiment, update its belief, and then perform another one and do this iteratively. So we did this again within the context of knocking down genes and measuring phenotype. And so here the goal essentially for our model was, can we identify strong responders? And so this is another screen in T cells, and we wanted to identify what are perturbations that lead to an overactivation of a specific pathway. And so what we do is the model makes its first guess of, okay, so you should try perturbing these 30 or these 60 different genes. That experiment is performed. You get a hit ratio, which tells you how many of the predictions that you made actually caused a strong effect. This information is fed back to the agent. The agent processes it and makes another prediction. And then that results in another readout. And so we keep performing this over multiple steps. And we see that over time, the model starts to pick more and more interesting hits or interesting perturbations. And we compared these predictions to, say, simpler approaches like using Bayesian optimization or using random baselines. And we found that actually our model was doing quite well across different data sets. Again, here, a lot of this was focused on T cells, on immune cells, because they were overrepresented in the data that we used for evaluating the model. But it's very encouraging to see that the model is doing something better than essentially kind of randomly picking genes, or even better than if you were to make use of kind of a Bayesian optimization approach. And then when we do a bit more deeper analysis, we actually find that these agents are able to make use of both the information that you provide as part of the prompt, as well as the experiments that they see. And so this is very encouraging. So for instance, here, the black line corresponds to a random baseline. The purple line corresponds to if you didn't tell the agent what you were doing and it just kind of saw every experiment without any context. Initially, it would start off pretty weak, but then later it would start to figure it out. But if you told the agent like, oh, here's the biology I'm studying, this is exactly what I want to do, it starts off pretty strong, and then kind of it keeps it up. And like, so this idea of like giving the model both the background information on what you're doing as well as the experimental data results in kind of the strongest results that we've seen, which is very encouraging. And it shows that the model has the ability to reason over different pieces of information. All right, and of course, this is there's a lot of people in at the frontier labs that are very excited about the potential of using these agents for designing experiments. In fact, the co-founder of Anthropic actually said that we shouldn't even be thinking about AI as just a form of data analysis, but instead we should just think of it as a virtual biologist that performs all the different actions that we do as biologists, and that should be able to really accelerate our ability to discover new therapies and make progress in research. All right, so yeah, this is the end of my talk. So thanks for listening. I just for a summary, I walked you through 4 different projects to talk about State, the virtual cell challenge, and also how we're making use of agents for curating and generating new data. Thank you so much for your attention. I want to make sure I acknowledge everyone in my group, in particular the people with asterisks against their names. They were lead contributors to the State model, and other collaborators at Stanford. If for any of the papers that I presented today, you can follow the QR code to read more in detail. Yeah, and happy to take questions now, or if you want to email them later. Yeah. Oh, sorry, I didn't see it. Yeah, yeah.
So I wonder, and I need to read the paper to get a better sense of the details on some aspects of the way the state model works, but so I recognize you're training in the optimal transport setting. It also seems like there's some similarities there to looking at this as a metric learning problem, where the choice of the stratification you're doing is sort of akin to batch mining, of identifying what things to populate your batch with in order to make the loss learn something meaningful and interesting as opposed to something that's sort of just noise. Is that a perspective that resonates at all, or is that something that you guys have thought about, or am I totally off base here?
No, that's actually a really cool way to put it. Yeah, for sure. I think when we do kind of align the covariates, we make it in some sense more meaningful for the model to learn variation because we've already corrected for the obvious variation. So yeah, I think that is actually an interesting perspective. We don't have any metric learning style losses. So like a triplet based loss, for instance, we don't have those in the model at the moment. So maybe we're not directly making use of set to set variation as a kind of loss. But I'm sure it's learning some aspects of that because we also tried this experiment where we artificially put in very heterogeneous sets of cells. So like you put in a different cell type, two different cell types. And then we actually do see in the attention patterns, there's like this like block pattern. So it's kind of learning to detect that. So yeah, it's an interesting way to frame that. Yeah.
Thank you.
Yeah. Yes.
I'm curious about like kind of the last pages where you discuss using agentic AI as a virtual biologist.
Right.
And you briefly went over how you broke down workflow, multiple agents doing different stages. I'm curious, how did you approach like building that out and deciding how what tasks that were going to be done?
Okay.
Yeah.
I think you're talking about.
Model.
You mean this thing?
Yeah.
Yeah. So yeah, no, that's a great question. So it's funny. So this was led by Nick. He's really into agents. He was the first author. I think when you there's a really nice tweet. I think I have it in here. Which it's a hidden slide. It's this one. So it's like me using LLMs for personal fun projects. Wow, this thing is such a genius. Why do we even need humans anymore? But then when you try to deploy them in the real world, it's like, oh my God, why is this thing so unbelievably stupid and dumb? And it's really exactly this. Like you start off thinking that, oh, you can just feed this to GPT and it'll figure it out. But there's so many possible mistakes you can make. Especially like and just being able to the way that you parse the text, and then the way that you map specific elements of that text to known vocabularies and known ontologies. And so because of that, what we ended up doing was we had multiple levels of agents that would perform similar tasks, but like check each other. So like you have here, for instance, I don't have the full hierarchy. I think we have it here. So like here's actually the full hierarchy. So you have like this overall supervisor that manages the lower level agents, and you have these lower level agents that are doing like specific things. So you have a find data set agent whose only job is to like find data sets. You have another agent that is focused on the eSearch attribute. And then and every supervisor kind of like checks the output of the agents that report to it and make sure that they're not making a mistake. So that it's kind of funny, but also cool how like these little agents are like checking each other, and then finally the response you get is like much more validated. So that's kind of how I think the system keeps getting larger. If you're interested in looking at this in more detail, we use a package called LangGraph. So if you may have heard of LangChain, it's essentially I think it's by the same people, but it's looking at a graph representation of agent workflows. Yeah. You had a question first, yeah. Sure.
I'm not sure if I get it correctly that essentially the generalizability is very great at it outperform the other model.
This one? Or you mean like
the external data sets?
Right.
Match the HPGs from your states to the external data sets,
yes.
or just using the model coefficient?
Yeah, we used HPGs, yes. And we matched the same gene sets. I should be clear that this is only looking at one metric though. So like if you look at we had like 5 or 6 different metrics. So on the others it was kind of mixed. because it is a difficult task if you go zero shot. But if you just start predicting like this metric, sorry, we call where'd it go? Like this metric, the effect size, which is like an aggregate effect across all genes, that tends to be kind of robust and it's like consistent across data sets. But if you look at individual genes, it's like much harder. And there we found that the noise between different data sets would overcome the predictive performance. Yeah. Yeah. Yeah.
The final results for virtual cell challenge, but like I know that since like for AlphaFold, like the protein database was founded in 2002, so it takes decades to have such a model like AlphaFold 3. So how long do you expect it to have like more to have enough data or highly sufficient model that can achieve as high as the accuracy rate as the AlphaFold 3 as like for perturbation prediction?
Yeah, no, that's a great question. I definitely think the bottleneck right now is definitely data and data quality. So because of that, I think it will be kind of a longer timeline. If it were just a question of, oh, we just don't have the right model, like you could probably make the case that, you know, it might be a shorter timeline because it's easier to iterate on model architectures. I think there's a lot that needs to be done on data. We just saw that ourselves when we put out the virtual cell challenge data set. It was just for one cell line. It's just like 300 or less perturbations, but it was already like, you know, showing much stronger and consistent signal compared to almost all the public data sets out there. And I think a lot of the labs are beginning to realize this, that we're not going to make any meaningful progress in the single cell space until we really seriously clean up the data generation process, you know, standardize the processing steps. And so BaseCount is an important step. We're now actually collaborating with other academic labs to even standardize the way in which the data itself is submitted to SRA. And, you know, so I think there's a lot involved on the data end, but it's great to see the momentum in the community. So at ARC, we have a 1 billion cell project where we want to, you know, generate 1 billion cells worth of perturbation data within the next couple of years. I think Chen Zuckerberg initiative also, they're also doing something, they're also trying to generate a lot of cells. So I think that will really accelerate the pace of progress. So definitely a decade is probably more than a decade, I would say. Yeah. Yes.
Question one for me, right? So are you working on isoform level configuration for the SRA BaseCount data sets? Isoform like usage for them.
Yeah, we do get that information. Yes, we do have isoform level information, and we've not done any deep analysis on it, but that's definitely one of the perks of our approach. Yeah.
Second question, I think another important perspective of perturbation experiment is how you define the readouts, how the phenotype is different. Do you have any insight on how to standardize that? Or because cell maybe like checking exhaustion or
Yeah, that's a good question. I mean, we work with, so transcriptomics is easy because, you know, you could just say, oh, pull out whatever you want from the gene expression. For us, because we're trying to be more unbiased, I think we're not focusing on any one specific readout. So at the moment, gene expression has been perhaps the most expressive one for us. I would say if we had to go deeper, I would be interested in bringing in some proteomic panels and like getting some of that information. Maybe some amount of imaging would be useful, might be overlapping with proteomics. Yeah, it is possible to train your model specifically for a phenotype of interest. You might see better power to predict effects. Yeah. Yes.
So kind of just follow-up question is I'm very impressed with what you have in the data set for gene expression so far, but you mentioned earlier that it's possible to curate data sets for proteomics and cell viability and all these other things. And is there what efforts are currently being made to incorporate those data modalities, if any,
Yeah, there are definitely efforts on the imaging front, which I've heard of, because I think imaging tends to be cheaper to generate even than transcriptomics, especially pharma companies have a lot of capabilities there. On the proteomic side, I think single cell readouts tend to be more expensive. There's more, I think, development that needs to happen on the assay and the technology side of things. So I'm not really synced in with what's happening there. I don't know if it's happening at the same scale, but there is a lot of interest in adding proteomic panels. I think definitely the vast majority of interest has kind of been more on the transcriptomic side, just because of the easier scalability. So I don't think there's anything at this scale, except maybe in imaging, I would say, at least to my knowledge. Yeah.